Thank you, grade 12 students. It has been a wonderful year meeting and having quite a lot of sessions that we interacted. I want to wish you all the best in your academic journey as you go forward. Just keep on working hard. I know that you can do it. And just check and also sign in your brothers, sisters, and every other young family related people who are also in Matric. As you all know that Matric Hub is actually one of those best sites for your life sciences. This is a genetics center. I guess you are just going to enjoy as we are going to dissect paper 2 here. Paper 2 is just going to be so interesting right now as we are going to read and prepare for paper 1. Please, I told you that the video for paper 1, which you are writing on Friday, which has been posted already, is actually an interesting one. Please go and check on it. Go and check on that. Don't make mistakes of just ignoring. We are here to coach and mentor you. We are here to advise. We are here to guide you. You will never go wrong. Just keep connected. All this that we are going to be doing here is for the best of your passes, not to struggle as you go forward with your studies. So please keep connected, and I really wish you all the best. As you can see, this one that we are going to be working on today is quite an interesting one. You shall see that all the questions we are going to explore and also relate and integrate every other related concept as we go through the paper. So what I want you to understand is you always have to make sure that instructions are read before you start writing. Read instructions of the paper as a whole, instructions also per section, and ensure that you follow and you don't make mistakes. So question 1 is actually various options that are given, which we need to find the best answer here. I was always saying that when we are given this question 1, which is actually showing alleles, we said these are variations of a gene. Alleles are a variation of a gene. As you can see here, the gene we are referring to, for example, here we are talking about the gene for height. We are going to talk about the gene for height, which is being explained under monohybrid crossing. It is a monohybrid crossing where the two organisms are only differing in one allele of a gene. So now what you need to look at, or what we need to understand here, is that in these circumstances, I want you also to say that you remember and relate the concept of complete dominance when we are talking about monohybrid crosses. We also have conditions and expressions of complete dominance, where the homozygous, we say this one is the dominant allele. We said it is the dominant allele, the dominant allele, and then we say the small letter is the recessive allele. So as you go through this, you know that the dominant allele always overpowers the recessive when we are going to look on the phenotypic appearance of an organism. So here now, we are given that the plant here is just tall, and we have got tall plants and short plants that are given in this regard. So the plant is dash. What is the expression of this plant when it is expressed in this condition? Homozygous dominant for height. We say that homozygous dominant class, homozygous dominant, it is whereby we have got both of the alleles are what are dominant. We say this is a homo that can represent a homozygous dominant. And heterozygous recessive, homozygous recessive also is just an opposite of what we have said. This is homozygous recessive when we look at that, which is also wrong. It is heterozygous for height, which is heterozygous for height is this one, which is exactly what is shown in the diagram. So the heterozygous for height is actually the right answer, which means to say that the plant is heterozygous, which is a combination of a dominant and a recessive. And then we are talking about incompletely dominant for height, that it is actually dominant. It is actually completely dominant. It is not incomplete, but we have got a complete dominant as we look into this, because the dominant allele will give the expression of the phenotypic appearance of this plant. So the answer is what? Is B in this answer. In this case, our correct answer is what? Is B. It is heterozygous, as you can see by the letters that I've given you there. Which of the following is not applicable to crossing over? Crossing over, we talk about this as the exchange of genetic material between homologous chromosomes. Genetic info is what is exchanged between homologous chromosomes. Homologous chromosomes, they exchange genetic information at the point of what? At the point of chiasmata. We said that this is between one stroke, 10 stroke is between. Please just take note of that. We use that in studying, but not in official writing. So which one is not applicable to crossing over? Crossing over, genetic information or genetic material is exchanged between non-identical chromatids of homologous pairs. It is correct that homologous chromosomes, or we are saying chromatids, they exchange the information, and then homologous chromosomes undergo crossing over in prophase 2. It is not in prophase 2 when we have crossing over taking place. uh, Crossing over does not occur during prophase 1. It is what? Early prophase. I mean, it takes place during early 1, and then the point at which genetic material is exchanged is the chiasma. It is correct. The process increases genetic variation. That is also correct. So we are saying this was the correct answer, which is in this case is not applicable. It is B. B is not applicable. B is not applicable. Crossing over does not take place during prophase 2. And then when changes occur in genes of sex cells, these changes, genes of sex cells, we are saying these are also, we call these also called what? The gonosomes. The sex, we are saying the gonosomes, which we can say the sex chromosomes. We can talk about them as also the sex chromosomes. So if they also, we are saying genes, okay, if changes occur in the genes of sex cells, the sex cells, obviously we are now saying in terms of the male and the female sex cells. So what happens if these changes occur? These changes, we are saying they lead to changes in the parent's phenotype. Changes may lead to These changes lead to changes in the parent's phenotype. They don't change the parent's phenotype because the parents are already there, and then we cannot have any influence that can come in that regard. Only affect asexually reproducing organisms. Changes in the genes of a sex cells, they only uh, affect asexually producing organisms. We are saying asexually, we are talking about vegetative propagation. That can also, it's a reproduction process that takes place. We are saying this is mostly in plants, which we talk about vegetative propagation. Vegetative propagation, however, we are saying always harmful to the offspring, can be the basis for evolutionary change. They can actually they can actually start to cause evolutionary changes. Evolutionary changes can occur as a result of what? Of changes in the genes of sex cells. So the answer is D, that they can actually start to cause changes in the evolution or evolutionary changes. Slight changes will start to occur in organisms, which by time can actually lead to formation of new species, speciation, and many other structural changes which can also occur in these organisms. It is a diagram that represents 4 different species of bacteria. We have got species A, species B, species C, as well as D, and then these are given like that. This which statement is correct? Which statement is correct regarding the survival chances of these species if there is a change in the environment? If there is a change in the environment, what would occur in terms of the changes of these organisms or the changes of these species of bacteria? So what we can see is that which statement is correct regarding the survival chances of these species if there is a change in the environment. Species A has the best chance of survival because it has the most variation. We are saying we have got variation. There is variation or the differences of the organisms. We can say this one can be, we can say it is black. We have got this one which is white. We have got this one which is also spotted or striped. So we have got variation there. In species A, we have got only black and striped. In C, it's only white, as well as in this one. So I would want to agree, believe that species A has the best chance of survival because it has the most variation. It has the most variation, as you can see. Variation in species A is big compared to other species. So A can be the best answer. That variation is mostly appearing in what? In we have got the most variation in A, and then the survival chances will occur because we say that survival is also affected by the variation chances or the variation that can be expressed within a population species. So the more variation that is there, the more the chances of survival, because given whatever condition that might cause or that might affect those organisms, what we can see that is that they cannot be affected equally the same because they are not the same. They are totally different. So we have got survival of the fittest being well expressed in what? In species A. The relationship between genes and alleles is that the relationship between genes and alleles. Genes and alleles, in terms of when we look at them, we must know that each person contains 2 alleles for the genes of one characteristic. The genes are different forms of the same allele. Genes can only have 2 possible alleles. Alleles are the phenotypic expression of a gene. So what we need to understand is that the relationship is that each person number 5, each person contains 2 alleles for the same gene of a characteristic, which is very correct to say that answer is B. I'm sure that you see that we talk about the homologous. I mean, we talk about the homologous chromosomes to say chromosomes, they appear in what? We talk about the pairing of genes. So therefore, each person contains 2 alleles of the same gene for one characteristic, which we can say that a person can be tall or can be short. It can be a certain complexion and another complexion, which would also strike an expression when it comes to the inheritance or the succession of that characteristic between the dominant and the recessive of the of the parents involved in terms of reproducing the next generation. So the diagram below represents the 4 limbs of 4 animals that all contain similar bones. So these diagrams are given here. We have got, I'm sure that you remember this can actually be a good one, which we also once discussed in previous presentations when we're talking about evolution. The the wing of a butterfly versus the forelimb of a whale, and also the human and also the cat compared together. So now what the question is saying is what is the question saying? We are given that the diagram that is below, we have seen it. So the question is saying what? It's saying the structures are structure one, the structures are homologous. The structures are homologous. The animals do not share a common ancestor. The four limbs of these animals have the same function. The structures arose due to modification by descent. I'm sure if some of you are actually still following and you are actually connected, as I believe you are, with regards to evolution, is one of the good and best presented chapter and section in on Metric Hub, which I am sure that for those who are connecting and following, life is simplified. So, so we are saying the statement above apply to the diagram dash. The statements above apply to diagram dash. So in terms of those diagrams that we have seen, these diagrams, we are saying number, it is, they apply to diagram 1. We are saying the structures are homologous. The structures are homologous. That is true to say that those structures are homologous. We say that they are homologous, which means that the structures arose due to modification by descent, which can actually be correct to say C can be correct. Modification by descent. I'm sure that if you go to Metric Hub previous evolution videos, we can be able to understand and appreciate that these structures are homologous. They originate they originate from a common ancestor, which is the reason why they have got some similarities there, which are actually caused because of changes, which are caused because of modification by structural similarities, and that is letter C. A certain disorder is caused by dominant allele F, and the following combination should be considered. The following combination should be considered. This is a recessive versus a. This is we are saying a recessive, and it is caused. We are saying the disorder is caused by dominant F. So you can say an affected is caused by affected in condition 1. Affected versus unaffected. I mean unaffected versus affected. So this is an affected person or an affected individual, if we look at on this one, and then this the second one is what is affected person. So if ever we see a combination like that, we need to understand that if it is recessive, it's unaffected. If it is this, it's affected. So it is unaffected versus affected. It is affected crossed with what? With affected. It is affected crossed with affected. So what is the question saying? This is affected versus unaffected. I mean affected because it is caused by what? By a dominant F. So which of the following combination of crosses can result in some offsprings without the disorder? Where can we have offsprings who cannot have the disorder? Where we can have the offsprings which cannot have the disorder. Number 7, we are saying which of the following combinations of crosses can result in some offsprings which without the disorder. So if we can look at that, we can say that if we say number one, it is where some of them are having are not affected, because we are seeing that when we further cross these questions class, you can please prove them, do the calculation, prove them. You can do the calculations. So then you can see. I can just want to demonstrate to you one simple one, which we can say that if we take first one, it is unaffected. It is unaffected crossed with what? With affected. So if we can cross that, we can say that we are going to have gamete. We are going to have gamete F, and then is crossed with this is we said is dominant and recessive. This is dominant and the other one is recessive. So we are going to have the dominant F, and then we are going to have the small f. So if we can cross this like that, this is going to give us ff, and we cross this with that, it is going to give us that. So this is unaffected, and it is affected. If you can do that, we can be able to prove. So we are saying one can be able to give us unaffected individuals which are not affected or without the disorder. So one is correct that it can give us unaffected. So if we can work it out from my observations, as I can look at this, if ever we have got a combination which have got we are saying a combination which can give us possibilities of recessive f crossing to each other. Therefore, we can be able to see that that can cause us what can give us unaffected individuals. As you can see here, we have got this f. It can cross with this f. We are going to have small f. So this is also unaffected. We can have unaffected here. You can also have unaffected there, but in any other cases, we don't have the chances of having small f's being crossed together there. So what I'm saying is this can prove to say that A can be the right answer, which we can have unaffected, and of course others are affected. We have got one unaffected and one affected. Same applies also on number one. One is to one unaffected and also one affected. So we have got chances that can be proven. This question, you can prove them by working them out as I'm showing you here. You can follow up on what? On question, on the diagram of genetic flow diagram that I've worked out, this one, which actually can show you how is this thing being worked out, or how can we have such an expression. So our answer is A in this case. Our right answer, I'm saying you should know that the answer is what? Is A. So question 1.1.80 and to 9 are based on the diagram below. They are based on the diagram below here. We are given these chimpanzee, we are given the Homo sapiens and Homo habilis. All these are given there and millions of years ago, which we say that you should know that this can be given in small, in abbreviation MYA, million years ago. So approximately how much longer did the trologites exist compared to the closest relative? Compared to its closest relative, how much longer? So here in this question, how much longer did the uh, troglodytes exist compared to its closest relative? We need to look for that. We should look for that on the diagram. We are saying this one is the one that we are looking at. The chimpanzee is the one that we are looking at. And then how much longer did it exist compared to its closest relative? The closest relative is what? The closest relative of this one. We can see that these are coming from here, and then they are coming. So therefore, their closest relative is this, which can give us the Australopithecus afarensis is the closest relative to that one. So what we need to see is that we're going to show the difference in years, how much different, or how long did each one, how did they live differently. So as we can see here, we are saying the difference, you are going to do the subtraction here. How much longer did the Pantrodites exist compared to the closest relative? So now, if you look at that, this one, it lived up to 5 million years ago. It lived up to 5 million, we can say 5 million years ago or more than, I don't know, it can be 5 million years, point something, 5 point something million years ago, the chimpanzee. And this one, it actually existed. Uh, it actually existed for something like We can see this, it came from here and then up to there. So we can maybe try to see how much longer. So it is one point something to 1 point, maybe 1 point. This can give up to something to 1.4 from 5 million years ago. We can say 5 million minus 1.4, 5 minus 1.4. I'm saying this is a calculation. It existed 5 million years ago. 2 minus the closest relative is this one, which we are saying 1.4 million years ago. So if you subtract that, you can be able to find out that can give you approximately or something like 3.6. It can give us something like that 3.6 million years ago. So that is the thing. You are calculating the difference, means subtracting. You are subtracting class. You are subtracting. This has to be worked by subtraction. You subtract the difference. Subtract. That is what the question wants you to do. You check the time of existence of the two, and then you subtract. Subtraction is what you are supposed to do in order for you to get that answer. So we can see that can be we can say that can be 5 million years minus 1.4 million years, and then we can get that one. And then the next one is saying, how many genera are represented in this diagram? How many genera are represented in this diagram? If we can look at it, we go back to the diagram. If we go back, let's see how many genera are represented on the diagram. How many genera are represented on the diagram? We can see we have got how many of them? We have got the Australopithecus, and then we have got the Homo sapien, we have got that and that. So as you can see, I'm saying this can give you a four genders that are represented in this. It can give you something. You can see that there are 4 genders that are represented in the diagram. So the allele, you have to give the biological terms in question 1.2. The allele that does not influence the phenotype in the heterozygous condition, that does not influence in the phenotype of the heterozygous. This is what I told you from that first question. We said this allele is the recessive allele. It is the recessive allele, that this one, it does not influence. The influence which is caused to, this is gonna give us to IE expression of the word that is the dominant allele. It is the dominant allele influencing this, influencing the outward expression. The plant is defined as TO. This TO is what is the phenotype. This is the phenotype. So as you can look at it, that is to say that this TO is represented by what I said. It's represented by the dominant allele, which is the capital letter T. So the recessive one is the answer. Recessive is the answer here. The answer is recessive allele. The bond that forms between amino acid molecules in a protein, we call that that is called a peptide bond. When amino acid molecules are formed, they join together by a peptide. It is a peptide bond, and then a portion of a chromosome that codes for a particular characteristic. A characteristic is coded by a gene. Genes, we are saying it's a gene. It is a gene that codes for a specific characteristic, which is a portion of a chromosome. Remember that genes are attached on the chromosome, and they are the ones which give the characteristic of a certain feature or a specific characteristic that we want to look at. The biotechnological process that produces a genetically identical organism. I'm sure you remember types of cloning. We have been talking about cloning. I had an illustration of the reproductive cloning that I showed and demonstrated on the channel here. It is cloning. It is a biotechnological process which is resulting in the production of identical organisms. We produce an organism from the existing one. We call that cloning. And then type of variation where there is a range of intermediate variable phenotypes. This I have also explained that as on the channel again, I expressed it to say that that is called continuous variation. It is called continuous what? Variation. I have given you some of these diagrams, which I said that the continuous variation, it shows a spread of the graph. It shows the spread of the graph. That is to say, we have got a range somewhere from here up to there. This is called a continuous variation. And then the opposite is a discontinuous one, which we can say that the discontinuous variation, it does not have intermediate values. We can have a graph that can be just like to say that we can have a graph like this. This indicates this will show discontinuous. This can show discontinuous variation. This is a discontinuous variation, and then this one is indicating a continuous variation. So we have got a sharp graph that rise and fall. It can only have one or two intermediate values or intermediate phenotypes that can be expressed there in a discontinuous. So the study of the past and present day distribution of living organisms across the continents, we call that biogeography. Biogeography, we talk. This relates also to the distribution biogeography. It is called biogeography. Biogeography. It is the distribution of living organisms across the continent, which we can talk about the Galapagos finches. We can talk about the tortoise in various continents. We can talk about the baobab tree in Africa versus Australia. That is the distribution of living organisms across continents. So the scientists who argued that acquired characteristics obtained by parents could be passed on to their offsprings. This is acquired characteristics. This was a theory that was postulated. This was postulated by Lamarck. Lamarckism. Lamarck actually is the one who had to argue and try to defend the issues and scientific issues to do with acquired characteristics, which we are saying during the lifetime. We are saying these are characteristics which an organism develop during its lifetime. During lifetime, characteristics are acquired, and then we say they cannot be passed on to the next generation because they are not in the what? They are not in the blood. They are not in the genes. We can say that they cannot, but Lamarck is the one who argued on that. Let's go to the next question. We know question 1.3, it is all about, it applies to A, it is both or none or all of the above. So characteristics of an African ape skull. Characteristic of an African ape skull. it is actually African apiscal, that is it is prognathous, which we say that it is what? It is it has got long jaws, which results for it to be called what? It is prognathous. And then characteristic of blood group inheritance. Blood group inheritance, it is actually, we can say we have got multiple alleles, which is to say that we have got multiple alleles, that there are 3 alleles in blood groups. There are three alleles controls the inheritance of blood. We are saying they control blood groups. So they are multiple, and then we also have codominance, which is what? AB is a codominant blood type. So this in question 1.3.2, this can be defined as what? As both A and B. It is both A and B. Is correct to say both A and B, they are all applicable in this multiple, because we have got 3. That is correct. And then is in a cross between a bull that is heterozygous for long eyelashes and a brown coat color, and a cow that has short eyelashes and white coat color, the garments of the cow will be dash. You see, this actually, it has got to do with, we are saying, we can actually, I also gave you a dihybrid cross gamete formation. You should be able to give the types of gametes that can be formed in a situation where we say that the bull is heterozygous for long eyelashes and brown coat color, heterozygous for eyelashes. It means to say that if we are going to use L, if we're gonna use L, we can say E. I don't know, E is representing eyelashes, and brown, it is also that. So if you can look at these expressions, they cannot suffice in terms of the expression of the garments. Can you also please go and look into this? I did calculated these questions, or I have demonstrated the forms, types of garments that can be expressed with a given dihybrid combination of alleles. So it's none of these. The out of Africa hypothesis states that modern humans originated in Africa and then migrated to other continents, which is correct. Some fossils of the genus Australopithecus have been discovered in South Africa, and then we need now to understand these geniuses that were discovered, and the scientists who discovered them. Which scientists discovered this? So name the name, give the name of the scientist who discovered misplace. Misplace was discovered by it was discovered by Robert Broom. And then the little foot, it is actually it was discovered by Clark. The little foot was discovered by Clark, discovered that, which we also want to understand that Clark discovered that. And otherwise, we can also talk about Mutsumi. It is Clark or Mutsumi. So, state the fossil site where Littlefoot was discovered. The fossil site where Littlefoot was discovered. I'm sure you know the cultural heritage sites, which we can say the Littlefoot was discovered from Stakesfontein Caves. Stakesfontein Caves, and then give the species name of the fossil Karabo. Fossil Karabo, actually, it was actually Australopithecus sediba. It was Australopithecus sediba. It is the one that was called Karabo. And then name the genetic evidence that supports the out of Africa hypothesis. The out of Africa hypothesis, it is actually supported by what? By the mitochondria. We are saying genetic. in terms of the genetic, that is where the question is lying. It is mitochondrial DNA, mitochondrial, it is mitochondrial DNA, which is written in short as what? Which we say that when we write it in short, we say M, small letter, mitochondrial DNA is the one that is a genetic evidence that supports out of Africa hypothesis. And then the diagram below represents DNA replication. DNA replication, as we go to exam my class, my students, can you please not forget that there are certain key areas of concern which you cannot go and you cannot be ready to answer that. DNA replication is one of the main subject areas which you should not go to exam without understanding it. So we are given DNA replication here, which is given this question. And the question is saying, during which phase in the cell cycle does replication occur? It occurs when do we have replication occurring? Replication, it actually occurs during the interphase. I have answered this question also severally on the channel. Interphase is where we see replication taking place. And then the structural state, the natural shape of molecule E. Molecule E states the which is this whole diagram. This is a DNA double helix. So it's saying the natural shape, it is actually a double helix. It is actually a double. It is actually a double helix. And then give the letters and name for each of the following. The letters and name for each of the following. A monomer, which is a building block of DNA. The building block of DNA. DNA is made up of what? Of nucleotides. So nucleotides, we should look on the diagram which one represents the nucleotide. Nucleotide, it is a combination of B and C as well as A. Nitrogen base, sugar, and also the what? The phosphate group. So it is D that represents. D represents what? Give the letter and name. It is D, the letter, and it is what? Nucleotides. And then a sugar in DNA. A sugar in DNA is actually deoxyribose sugar. The sugar in DNA is deoxyribose sugar, which is actually a deoxyribose sugar is the sugar in what? In DNA. So saying the sugar in DNA, which is deoxyribose sugar, which is represented by C. We can see that the sugar is represented by C, which is deoxyribose sugar. C, deoxyribose sugar. And then the complementary base for the nitrogenous base in the complementary base for the nitrogenous base in molecule D. If you see, we are saying the complementary base for the nitrogen base in molecule D. Molecule D, we are given that molecule D, the complementary base, the base that we are given here, it is what we are given, thymine. So we say thymine, it pairs with what? With A. Thymine pairs with A in terms of DNA structure. A pairs with T, so it is what? It is adenine. It is adenine, which can also be written as A. Two skeletons are shown in the diagram below, and we are given the skeletons here, diagram A and diagram B, which are given. We are given this 1.6, and then the question is saying identify which of the diagrams A, B, or both belongs to the Homo sapiens and Homo habilis. So which of these belongs to the Homo sapiens? The Homo sapiens are actually the modern men which are bipedal. We are saying the Homo sapiens are bipedal. So you can see diagram A represents the Homo sapien. It can say Homo sapien is bipedal. We can see that is bipedal, which is actually diagram A. And then hominids, we are saying which of these actually represents the hominids. The hominids, we are saying the Homo sapiens, they also belong to the hominidae. So we are saying it is both A, it is both A and B. It represents the hominidae, which is A and B. Describe 3 characteristics of an organism skeleton that allows for bipedalism. This one is also, guys, you can refer to Metric Hub, also refer to the channel as well. Refer to channel. We have got a full video that explains. Refer to the channel. We have got a full video that explains bipedalism. So one characteristics of organisms are skeleton that allows for bipedalism. One, we need to understand that we can talk about the wider and shorter pelvis. Bipedal have got shorter and wider pelvis. And then two, we can also need to understand that we have got a more forward position of the foramen magnum. More forward position of the foramen it has got a more forward position of the foramen magnum, which would allow the vertical seating of the spine. And then lastly, we can also talk about the S-shaped vertebral column. The S-shaped vertebral the S-shaped vertebral column. These are the characteristics of bipedalism that I think you need to always take note of. Characteristics of bipedalism. These are the ones: shorter pelvis, shorter and wider pelvis, and also the more forward position of the foramen magnum, also the S-shaped vertebral column. The main characteristics. State the characteristic of the socket of the skull that allows binocular vision. There's binocular vision. Binocular vision, we are saying ability to see with both eyes. Seeing with both eyes is what we call binocular vision. So what is that? The eyes are actually in front of the skull. Eyes are in the front of the skull. Eyes are in front of the skull, so therefore it allows binocular vision. Therefore, that can allow binocular vision. We are saying, as you can see, they are able to see more forward, as to say the eyes are in front position. They are in front position. That is, the eyes are in front position. Hence, we are saying that binocular vision is supported. Question 2, it shows a partial DNA sequence of a human, and table 2 shows the amino acids coded for the different by the different mRNA, what called messenger RNA codons. So we are given here table 1 base triplet number, we are given 1 to 4, and the human DNA sequence TGT, ACG up to AAC, ACA, and we are given table 2, the mRNA codons, and then the amino acids that are coded by them. So we are given that V line, it is coded by these 3 codons, as well as many of them that are given, trisine and the leucine and so forth. The question is saying, the question wants us to understand, to say, name and describe the process in protein synthesis that is responsible for formation of mRNA. This process class, I always want you to understand that the process of mRNA formation, it is actually a transcription. You are asked to describe transcription. Transcription is what you are supposed to talk about. You are asked to describe transcription. Transcription is the formation of mRNA from DNA. We are saying the formation of mRNA from DNA is what we call transcription. Formation of mRNA from DNA is what we call transcription. So I have been describing this severally again, and I feel like for some of you who are following, it is quite overdone now. That in terms of the process of transcription, you need to understand that the double helix DNA, it actually unwinds, and then there is the double-stranded DNA, it actually unzips after unwinding, resulting in the breaking down of what the weak hydrogen bonds, they actually break to separate the 2 strands, and then 1 strand is used as a template to form mRNA. 1 strand is used as a template in the formation of mRNA, and then using the free RNA nucleotides from the nucleoplasm. The mRNA is complementary to DNA, and then we can actually have the formation of mRNA. I have given you that this, you can see a small portion of DNA unwinds, and one of the strands is used as a template, which I can maybe want to say that either we can further come back, and then it can further come and then go there. So this piece which unwinds, I can say that we have got this piece there which unwinds like that, and then we are saying the order of the nitrogen bases that are found on the mRNA, on the DNA structure defines, we are saying, using free RNA nucleotides in the nucleoplasm, the mRNA is complementary to DNA. So would have, if ever you see the formation, if ever you're gonna see a diagram like this, where you are going to find out that we have a single strand that will form here, that will form in between the broken strand, and don't forget that this is going to be inside the cell, and then it has to live through the nuclear pore. So we are going to find a nuclear pore, which we can say that it is like this, and then it is like that. So through this gap, you shall see that it can be given like this, and then it will come and lie there. This diagram represents what? Represents transcription, as I've narrated and explained it. How do you say that? Give reasons. Because we are seeing mRNA that is being formed here. We have got mRNA that is formed. After it is fully formed, it moves out through the nuclear pore, and then it is lying outside the nucleus. So we can say that that is what you need to take note of. But the explanation is like what I've given you already. Give the mRNA codon that is formed from the base triplet number 4 on the DNA sequence. Give the mRNA codon that is formed from the base triplet number 4. Base triplet number 4. we are saying give the mRNA codons base triplet number 4. This is the base triplet number 4 that we are looking at. Base triplet number 4. So give the mRNA, give the mRNA codon that is formed from the base triplet number 4 on the DNA sequence. So what is given here is that you are given, like I said, you are given A, C, A. So what is going to have is that you are going to have U, U, G, U, A, C, A will pair with what? It will pair with U, and there we have got G, on there we have got U. So you have got UGU that you can have being formed on that regard, which can cause you to have U, UG, UGU, UGU can be formed in that uracil, guanine uracil. Name the amino acid coded by the base triplet 2. The amino acid coded by base triplet 2. The amino acid that is coded by base triplet 2. Base triplet 2, we are saying base triplet 2, it is ACG. And then we have to look for that to say ACG is coding for which amino acid does it code for. We are saying base triplet 2, which amino acid is it coding for? So what happens is that base triplet 2, we are given that this can be, we can have that to say that it can actually represent to say base triplet 2, this is the DNA. So if it is in mRNA, we're gonna have that we are going to find out the corresponding in terms of what? In terms of mRNA. In terms of mRNA, which can give us to say we can have this can be given to say that in terms of mRNA, this can result in us having we can say UG is U here, is G, is C, UGC, 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 UGC is the is actually the is actually the codons that we can find there, is the mRNA codons that we can say UGC. So we look for UGC and identify the amino acid that it represents. UGC, 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 it is here, it is U, it is G, it is C, which we can say it represents the amino acid cysteine. It represents the cysteine amino acid. It represents cysteine. It actually represents cysteine, which you can see is the one that I've given you how you calculate it there. And then mutation caused base triplet 1 to change. It causes base triplet 1 to change so that it looks the same as base triplet 3. And then explain what effect this mutation has on the protein formed. This question is also one of the key areas that I've already many times given you and guided you that you should always be able to look at this and understand what happens to say base triplet 1, it changed to base triplet 3. Base triplet 1, this one, it is T, G, T, and then it changed to T, G, C. So when you look at that, we are supposed to identify to say when it is TGT, it changed from TGC, TGT to TGC, as that it changed like that. you should be able to understand to say, what are the effects of this mutation? Mutation is one of the key subject areas that I'm sure that you guys you are quite aware, and we have severally tried to explain and bring to your attention all these circumstances through which, or areas of concern which you can see how does that question change. So we should look at this, and then let's correlate the mRNA codons of TGT. What are the mRNA codons here? We're gonna see that this are the best. We are saying it can change to A, it can change to A, C, A. This is ACA, and then it changes to A, C, G. We are saying these are the codons here, the codons that are formed in this case, the codons that are formed. So what we need to look at is, does that affect how does that affect the type of amino acid that is formed? ACA to ACG, that's the change that we have. So what we need to look at is that we have got ACA, it changed to ACG, it changed to A, to it changed ACA, changed to ACG. So if we look at that ACA, do we have ACA here? ACA is this one, and then it changed to ACG, it changed to ACG, which is also the same. So here we still have the same amino acid being formed. We have got the same amino acid that is formed here, class. The question is, we are concentrating in this boundary. We are concentrating in this boundary that I want you to see, to say, where are we? What is happening here? So we are looking at this board in this box here. So we can see that there is ACG, ACA changed to ACG, which remains the same, giving us the threonine. We remained having threonine, what call amino acid. So in this regard, there is no effect in terms of the mutation. What we did, we therefore need to say is that we basically we just have to say that the base triplet, which we say that our base triplet are TGT, uh, it will, it will change to T to TGC, and therefore the mRNA codons, therefore mRNA codons will also codons changes, the changes to the changes, let me say the changes from ACA to ACG, which causes uh, for the same amino acid. So the protein will not be affected. That is what you need to see. I'm sure that we have seen the demonstrations which I was showing you here about how these changes are taking place. TGT change or change to TGC, and then obviously the codons will also change from ACA to ACG. But however, they are going to code for the same amino acid, the threonine. So the protein will not be affected. Same protein, since same protein is produced. That is what you need to understand. If ever the protein has changed, therefore, if ever, let's say that there was a new protein formed, you were also supposed you are only going to give a narrative of which it changes from where to where, it changed from which protein to which protein, or else also you change the changes in the mRNA codons, which changes from this to that. So that is basically what you need to do. You are actually giving a description of what you can see according to the changes that are taking place. The diagram below shows a cell during a phase of meiosis 1 and a phenomenon that occurs during the phase of meiosis, which may lead to chromosomal mutation. It can lead to chromosomal mutation. So we are given that is what we are given here. We are given that, and then we are given that question, and then the question which we want to answer here is that 2.1.1. At the end of meiosis, gametes are produced. That is very correct. Explain one other biological importance of meiosis. One other biological importance of meiosis. We are saying what is the other one? The other importance of meiosis. Meiosis actually it is importance. It actually it increases survival of organisms. I'm sure I remember survival of the fittest. It actually importance genetic variation. It actually allows formation of genetic variation. It allows actually for genes to be different. It actually causes various genes to be formed, which is very, very important. So we need to understand that it introduces genetic variation, which is through crossing over, and then also through crossing over genetic variation, it takes place, and also it helps to overcome the doubling effect of fertilization. It also avoids doubling effect by halving the number of chromosomes. The number of chromosomes are halved during meiosis. I'm sure that we are quite aware of that. That is one key and the important answer that we can give. And then tabulate the differences between meiosis 1 and meiosis 2. This can be just to say the differences between, you can just say the difference between meiosis and mitosis. Remember, meiosis 1, meiosis 2, it can also be the same as mitosis. So when they say tabulate, you are just supposed to draw, put it in a table form of the differences. Meiosis 1, and you say meiosis 2. That's what you need to say by in meiosis 1. This is also a repetition of one of the questions that we have done before. Here we say crossing over occurs here. Crossing over takes place during meiosis 1. No crossing over occurs here. no crossing over occurs here. We can also talk about in the end here, we have got two cells formed at the end of the division. In one, two cells are formed at the end of the division. And then at this one, we have got four cells are formed. Four cells are formed at end. And then we can also maybe say 3, the chromosome number is halved. And then in the other, in meiosis 2, it is constant. It remains constant. And then lastly, I can also say that one last point that I can give is that the chromosomes arranged in at the equator of the cell in homologous pairs. Chromosomes arranged at equator. They arrange at equator in homologous pairs. They arrange in homologous pairs in terms of meiosis 1 and then meiosis 2. In meiosis 2, what happens is that chromosomes line up at the equator of the cell individually. Chromosomes line at equator individually. They line at equator individually. In terms of meiosis 1, they are as homologous. There are two of them. So these are the basic differences that I want you to basically understand and perceive when it comes to this. Draw a labeled diagram of this cell in the phase before one shown above. So what we need to do is that we are saying you need to draw this diagram, which you are going to show that the metaphase 1 is what we are going to show here. So the question is saying diagram of the cell of the phase before the one shown. So we are saying before the one shown, so we are going to say that the title is metaphase 1. Metaphase 1. And then we are going to see that we have our cell there. And then we can have this and that. And then this and that. We can say this and that. And then we can be able to draw to say that the central Here we have got to say this. I've got a label with a red pen. This can indicate these are homologous pairs. These are homologous pairs. And then we can say this can be actually be the spindle fiber. We have the spindle fiber there. We can also have here, which indicates the centromere. And then we also have our central we can also say this one, a single piece of it is called a chromatide. That is just a single piece of the chromosome. And then obviously we can say, we are saying this is that, and here is the cytoplasm. So all these can be given in this respect. We can have that diagram just to say metaphase 1, and then you draw your diagram as like that. You draw your diagram as like that. Sometimes for exam purposes, I want you to know the functions of these to say the spindle fibers are there to move and adjust the positioning of the chromosomes in order to effect the process of cell division. We have got the cytoplasm, which is actually there in order to contain and maintain the shape of the cell. The homologous pairs are just the two chromosomes. And then we talk about the centromere joining the two pieces of the chromatids together to form a chromosome. Those are the things that you need to look at and understand. Name and describe the phenomenon that occurred in the cell above. When you look at this one, we say that the cell above, we say that, as you can see, we have got two of them here, which we can define that there is non-separation of chromosomes, non-separation of chromosomes that takes place, which they can also say non-disjunction. So the question is saying name and describe. It is non-disjunction, non-separation of chromosomes, which we can say that this is whereby what happens is that the chromosomes fail to separate during anaphase 1, a disorder which occurs during anaphase 1, where we can see homologous chromosomes failed to separate, where homologous chromosomes failed to separate. So what happens is that we are saying that the homologous chromosomes did not separate. It's a disorder which normally results, which we say that it normally occurs in trisomy. It is resulting in trisomy 21, which we see that you can see a 2N cell is being fertilized with an N cell, and then this will give us 3N, which we can say a trisomy. It is called a trisomy condition. If ever we can further explain it from there, if we can further explain it, that's how far we can go. Trisomy condition resulting in what? Resulting in Down syndrome. And then we can explain all those as you can go further if ever the question wants you to go more than that. But that is what you basically need to understand here. The 2N is the non-separated cell, and this is a normal cell, an abnormal and a normal cell. So this is a haploid, and this one is a what? Is a diploid. That is one thing that we need to understand as we go into details of meiosis class. So we are given that at the end of meiosis, in this cell, if one gamete containing 3 chromosomes fertilizes fertilizes a gamete containing 2 chromosomes, would this lead to Down syndrome? As you can see, we say that Down syndrome is caused by trisomy, but here we have got 3 and 2, so it cannot. No, it cannot lead to Down syndrome. And then give one reason for your answer. This is because this is not a human cell, as it shows there. The cell does not show one of human. And then as the zygote, as you can see, as the fertilized zygote will only have 5 chromosomes. It does not have 47. The zygote will only have 5 chromosomes. Not 47. 47 are the ones for humans. Not 47 chromosomes. Not 47 chromosomes. So this is therefore that cannot be causing Down syndrome, because extra cell is not a 21 pair. Extra cell is not 21 cell, 21 pair. That's what we can see in that regard. We are given rare cases, occurrences of babies being twitched at birth in hospital have made news in the past. Individuals' DNA sequence is unique with exception of identical twins. So DNA profiling is reliable of determining the parents of a given baby. Restriction fragment length, the polymorphism, or using the polymerase chain reaction, PRC, are 2 methods used in analyzing DNA profile. This is quite true. So we are given these, the hospital allocated babies as follows. Baby 1 was given to couple A, baby 2 to couple C, and baby 3 to couple B, as like that. That is what is given in this case, DNA profiling. So from the text above, state 2 methods used to analyze. From the text above, state 2 methods used to analyze DNA profiles. These are given class. I'm sure that you can see we are given the RFLP and also PCR. They are the two which are given there. So we can talk about the polymerase chain reaction. We are saying we have got the polymerase uh, we have polymerase chain reaction, and also we have got restriction fragment length. Restriction fragment length polymorphism, which is also RFLP, and also we have got the PCR. These are the two methods that can be used. And then based on the DNA profiles provided, which baby does, which baby or babies were correctly allocated by the hospital, which one was correctly allocated DNA profiling. I have answered this question severally class about DNA profiling, as we have seen that these we said that you should match the profiles of the child of the child to the parents. You should see the matching effect of the profiles to what, to the parents. So we are saying which babe was properly allocated. So if we can see, you see babe one, all of them, babe and daddy, they are, we can see there's a likeness. Two, baby two, if we can go there, if we can find out this, we can also find that, and then we can as well find all these. So what I'm saying is that in analyzing the baby correct allocation, it is all about matching the matchingness or the similarities of the DNA profile of this one. So we can see that this can be baby two, and then the reason is that each bar, we say that the reason is that each bar in the DNA profile of baby two matches with the bars of either each bar of baby two matches matches with it matches with the mom and dad, matches with a bar of both parents, either the mother and the father. So there is that reason there. And then provide one other use of DNA profiling, one other use, one other use, one other use, meaning to say other than that one that we have talked about, paternity. So it can be tracing missing persons, tracing missing persons. We can also talk about establishing, we can say identifying dead bodies. Identifying dead bodies, we can use DNA profiling. We can also talk about uh, we can talk about identification of genetic disorders. Genetic disorders can also be identified through that. And then we can also maybe talk about criminals, identification of suspects, which we say suspects in crimes. Criminal cases. Criminology is also one other way that we can use that. We are given in species of fly, Drosophila melanogaster, tan light brown body is dominant over black body, long wing, and dominant over short wing is given there, female and male. So tan body color and long wing length, black body color and short wing length. So that is what is given in this. So the punity diagram shows the cross between the male and the female of these as shown above. So we have got the possible gametes, that is the one and that one, and then we have got that, and then the question is saying, one 2.41 states why the type of cross involved in the above scenario is referred as a dihybrid cross. I have given you that it involves two, it actually because it involves two characteristics. It involves two characteristics. It is because it involves two characteristics. It involves two characteristics, which we said that the two characteristics that are involved there, we talk about the body color and the wings length. That is body color and wing length. The body color and wing length. And then state, okay, the formation of gametes with a different allele combination can be explained by Mendel's laws. State the name. Okay, name the two laws that apply here. We talk about the law of independent assortment. I'm sure that you remember this one. Law of independent assortment. The law of independent assortment and the law of random segregation. The law of random segregation. I'm sure these are the laws. I'm sure we have already also explained a lot about these laws, and then identify the genotype of the gamete of the male parent A, the genotype of the male parent A, the genotype, we say that it is supposed to be represented by what? It has to be represented by letters. It has to be represented by letters. So the genotype of the male parent A, we are saying that possible gametes. So A, we are saying that this can be, I have explained these ones quite a lot of time. How do we come genotype of the male parent A? It has to be, it is given that it is, we are given that it has got a recessive, it has got a recessive and a recessive in terms of the body, in terms of the color and the what, and the color and the color and also the length. So it is actually that it has to be what? Recessive. It has to be recessive, both of them. And then phenotype of the offspring labeled B, phenotype of the offspring labeled B, the phenotype of the offspring labeled B. So this, the phenotype of the offspring labeled B, obviously if we come here, this gonna give us to say that the phenotype is going to be, it is going to be, according to this, we have got the dominant, saying the color, we are given that is going to be species tan light brown body is dominant over black, color, and long wing is dominant over short wing. So brown is dominant over what? Also over black. So on this one, what we can say is that it has to be the dominant one is actually, we are saying this gonna give us to say that it's going to be it is going it's going to be a black color with long wings, black color with long wings, black color, and then we have got long wings. So it has to be black color and long wings. Genotype of the female parent, we are given that the female parent, the genotype of the female parent, it is actually given that from this it has to be long wings. It is said that the female is dominant over, we are saying Drophila tan brown is dominant over the black, which is dominant over black, and long wings. I mean long wings over short wings. So we are saying that it is Drophila tan brown body is dominant over black body. So T, we have got T is dominant over black body, and then long wing is dominant over short wing, which we say that again we have got L and small l. So now tan body color and long wing body. This is the female, which is given that the female is what? The female which we are given here, it is tan body color and long wing. So tan body color, it is the tan body color which is given here is that we can say that the tan body color, it can be tan body color is that one, and then it has got long wings. We can say the long wings is that. So in this case, what you need to understand is the combination of the alleles. This is representing the color, which is to say that we have got tan is what? Tan board. The can, which is tan body, and this one is indicating the length of wings. Which we are saying that they are long. Length of wings, they are long. So we can get now to say that our genotype of the female the genotype of this female is therefore given as to say heterozygous T and homozygous what L. Heterozygous T and homozygous L is what we can see in this case. It can be to say that we have got it has got tan body and long wings. This is the what? The genotype. Tan body, and then we are saying it is based on tan body and long wings. This, I'm just qualifying my answer. It's not being asked here. I'm just qualifying my answer for you to understand, to say why did we say that answer? It is because of that, to say that our tan body is what? Is that one, and long wings, it is what? It is long wings is shown by the LL. And then when we combine them together, when we put them together, then it gives us what? This will give us, which we are giving as the answer then. The diagram shows two models of evolution based on the rate at which it occurs. So we are given this. I'm sure you remember the two modern theories of evolution, the punctuated equilibrium and gradualism. I'm sure you still remember these two theories, punctuated equilibrium and gradualism. So there is now a question: give the letter that would best represent gradualism, and then Eldridge God's explanation of evolution. So we are saying give the letter A or B that would best represent A. Give the letter that would best represent gradualism. Gradualism is actually represented by what? It is actually represented by B. As you see, gradual, slow changes over long periods of time. We are saying this is what? Gradualism. It is gradualism. It was what? I mean, it is actually shown by B, and Edric God as explanation of evolution, which can actually explain the opposite, which is what? Which is A. And then 2.52, name and describe the model of evolution shown in A. It's the model of evolution in A. This is what? Punctuated equilibrium. This is punctuated equilibrium. And then now, that is what you are given there. So name and describe the model. So you are just going to explain punctuated equilibrium as to say evolution involves long periods of time where species do not change. In A, it involves long periods of time without any change. So we are saying that evolution involves long periods of time without any change, which actually we need to understand that this is a theory of evolution, alternative or opposite, which is opposite, to uh, we are saying which actually it actually alternates. This alternates with punctuated alternates with short periods of time. It alternates with short period of time. It alternates with a short period of time, which I'm saying that where rapid changes occur forming new species. Short period of time where rapid changes they do occur forming new species. New species by natural selection. By natural selection. That is what you need to understand in terms of punctuated equilibrium. But we can as well go to our channel. It has been severally explained and was given such a lot of attention. And then name the type of fossil that supports the model of evolution represented by B. We said that the type of, this is gradualism, the type of fossils that supports the change, the model of evolution. This is actually supporting that the changes that are taking place from the start to the end. To say we are looking at the evolutionary changes, evolutionary changes that are demonstrated in this case. So how are they demonstrated? We are saying we have got what is called a transitional species. A transitional species is actually being used in this case. Transitional species, which has characteristics of both, which shows characteristics of both. It is an intermediate species. So now what you need to look at is the fossil is the transitional. It's actually a transitional fossil. Transitional fossil. It can show us how changes have occurred. It can show us how the changes were occurring. That's what you need to understand with that. And then question 3 is domesticated crops have been transformed over thousands of years and now look vastly different in comparison to their relative world. Maize belongs to genus Zea, which includes wild teast, and then domesticated maize, Z maize. So we are given that these are the Toscent produces a cob with only 6 to 12 kernels in rows protected by a hard outer covering. Modern maize produced by farmers have a cob consisting of 500 or more exposed kernels in as many as 20 rows. The diagram below shows the wild Teoscent and the domesticated maize cob. So name the process through which the humans produced the modern maize. Modern maize is actually produced through artificial or selective breeding. It is artificial selection. Or we can call it selective breeding. That can also be called selective breeding. And then state one negative effect the process named in question 1 may have in species. We are talking about this artificial selection or selective breeding. It actually decreases biodiversity. It decreases the biodiversity, variety of organisms in the natural ecosystem. It decreases. And then 2, we can also talk about it that it also decreases the gene pool and genetic variation. Reduces genetic variation, or we can say gene pool. And then it also, maybe number 3, say that it increases genetic disorders. And again, we can also say that it may cause mass extinction. Organisms can die in many numbers because of that. There is no natural selection. Mass extinction can take place because of artificial or selective breeding. And then describe how the process was carried out on maize, how the process was done. Actually, farmers, in terms of that, what happens is that farmers do select. They do select the maize with the maize plants with more kernels. Select maize plants with more kernels, and then these were interbreed them. They interbreed them, and over thousands of years they developed modern maize. Over thousands of years. They developed the modern maize. So they chose the maize seed or the maize plants with the favorable characteristics, which we can say that they can have more corns, they can have more rows, they can have bigger ears or a bigger what, and then these were interbreed, and then over time they now managed to develop the modern maize. And then explain one economic benefit that producing the modern maize cob has to farmers. It actually high yields are produced because you have heard that more rows and more maize cobs are there, more maize grains are there. It results in higher yields, production. It can also talk about more profits, more profits are made. Those are also advantages that we can put in that respect. And then we have got an isthmus is a narrow strip of land that connects two large land masses and separates two bodies of water. We are given that the isthmus of Panama is a narrow strip of land that joins North and South America. Scientists believe that this strip of land formed 3.5 million years ago. Caribbean Sea is warm while Pacific Ocean is cooler. The porkfish genus Anestoremus can be found on either side of the narrow strip. However, when scientists placed male porkfish from the Caribbean from this and that into the same fish tank, they would not copulate, so they could not interbreed. They are now considered to be two different species. So define the term species. We are talking about species as a group of organisms. It can be a species. We can define that as a group of similar organisms that are capable of interbreeding and producing fertile offsprings. Group of organisms that are able to interbreed and produce fertile offsprings. If it is a species, it means when they are interbreed, they can produce a next generation. A next generation can be produced. Name two reproductive isolating mechanisms that may have prevented the two species from copulating. This can be actually due to prevention of fertilization. Prevention of fertilization. It can be prevention of fertilization, which means to say that fertilization was not able to be given chance. It can also be species-specific courtship behavior, a different courtship behavior, which we can say that is called species specific courtship behavior. That can also be the reason, and then it can also be because of differences in breeding times of the year or a season. This can be some of the reasons that can also cause that. This can be some of the reasons which can cause, we talk about reproductive isolating mechanisms. There are these ones that we can give, which can also stop breeding. And then based on the scenario, describe the speciation of porkfish, geographic isolation. Please, this one, please refer to channel. Please refer to channel. Talk, just search geographic isolation, speciation. Please refer to channel about on this one. It can give you a geographic isolation to say one single population is divided into two or more groups because of geographic barriers, which we gave the different types of barriers. They can be land, it can be water, it can be a mountain range, it can be a golf coast, it can be a desert, whatsoever it is. So the two population species undergo each one its own reproduction independent of each other, preventing gene flow, and then these two populations will become genotypically and phenotypically different. So there is a diagram that shows these descriptions that I'm explaining right now. We are given this question here, which is the ichthyosis is a skin disorder caused by mutation of gene carried on X chromosome. Disorder results in dry skin due to deficiency of an enzyme. Individual B has got a genotype that is given that one. So we are given that the unshaded shapes here represents unaffected and the shaded one represents affected. This is also quite an interesting topic which we have done severally again. 3.3.1. What you need to say is that this is quite what you call a repetitive question that we have done several times. We have done it several times, and then I'm sure that you can relate very well. You can relate very well to these questions, which we can come to, and then name the type of diagram that is shown there. This is a pedigree diagram. It's a pedigree diagram that is shown. This one, it's a pedigree diagram. This is a pedigree diagram that we can see here. And then provide the possible genotypes of individual A. This one, the possible genotypes, it's a she. It's a she. So what we are saying is this: the possible genotype of this person, she can be unaffected or she can be a carrier. So there are possibilities. The possibles is being genotypes. She can be, let's say that she is unaffected. So we can have our XX as to say that she is unaffected, or we can have that she is a carrier, which we can have our XX, but with a what? With a heterozygous T. That is what it can be there to say that this person, we can have possibilities of that. Remember we said, if it is like this, a homozygous T, we said that is an affected female. And then we say that the other one, which is, it is showing to say that is a what? Is a carrier who cannot show that she has the disease, but can only be seen in the children that she's gonna produce. So explain the relationship between the inherited genotype and the phenotype of individual C and Mendel's law of dominance. This we can also talk about issues to do with individual C Individual C, this one is actually an affected male, is an affected individual, is an actually affected male. Individual C is an, okay, C is this one. Okay, we are saying this is C. Our C is this one. So this person is actually an affected, or we're saying she's a career mother because she has got an affected son here. This is a individual C is a career mother. As you can see that she is a career mother because she has an affected son that we are seeing there. Is a career mother, who has got this son D. Is a career mother as she gives rise to son D. Individual C is a career mother because she gave birth to an affected. We have got an affected son there, who is D, affected son, who is D there. And then this, she may have inherited she may have inherited the allele from a mother. She may have inherited the allele from a mother which was also covered by the dominant allele T, sorry. So there was a dominant T that might have been covering this one. So a phenotyping may show a being unaffected, as we say that, as we have seen there, but it can only show in the children. Can only be expressed in your children. That is what we can see there. This question 3.34, it is one of the questions that we have seen severally again. It is that male are more likely to be affected by the disorder than females. It is because males have got only, we are saying the diseases that we are talking about, these are sex-linked diseases, or we can see that they are carried by an X chromosome. So what we need to understand is that men or males only need to inherit They only need to inherit one recessive allele. Male or female. Needs to, then well, female needs to inherit two alleles for them to be affected. For them to be affected, they need to inherit two alleles. That is what you can see there. So male can easily be affected because they only need one allele. Therefore, they can actually get affected easily. And then next question is saying, Nala and Luke are farmers of the same species of small fish. Nala breeds homogeneous yellow fish, while Luke breeds homozygous blue. In an investigation to determine the dominant color in this species of fish, they allowed for the yellow fish to breed only with the blue fish over 5 years and collected 100 offsprings each year to observe. So the table shows the average results of what? Of the offsprings. The table shows the average results of the what? Of the offsprings. So now, what is given here is that we are asked here to see to it that we are now supposed to identify and see the questions that are arising here, saying name the type of dominance shown in this investigation. I've been giving you previously that these investigations are just ways in which we have also understood and given that if we talk about homozygous or we talk about a monohybrid, we talk about complete dominance. So the table shows yellow average number of fish over 5 years. Yellow is 20, green 65, blue 15. So we can see that this is what we can talk about complete dominance. So we have got, so we say that these types of dominance, as it is shown there, these are the things. So as you can see, I want you to look into this and then you'll be able to see. We have talked about the ways in which the results of an investigation can be improved. That has been answered also in the previous question of paper 1. We have answered that one as well. So I trust that this has been well done, and because of this time and also this network thing, we can continue it from here. Thank you very much and have a beautiful stay. Let's keep following and connected.